Hello everyone, myself Nerityajeet. Welcome back to my YouTube channel. I hope you all are doing great, guys. Today in this video we are going to learn about respiratory balance sheet and amphibolic pathway. Basically in respiratory balance sheet, we assume certain assumption for proper oxidation of glucose molecule to get 38 ATP molecules from it. So assumption are that first assumption is that all this pathway is held in an ordered manner. Like firstly, glycolysis happens, process of glycolysis happens. After it, link reaction, and after link reaction, there is Krebs cycle, and after Krebs cycle, electron transport system or electron transport chain happens. By all this pathway, we get energy. So like glycolysis, we get 8 ATP molecules, and by link reaction we get 6 ATP molecule, and by Krebs cycle we get 24 ATP molecule. Total ATP molecule is 38 ATP molecules. And the next assumption is, that the NADH formed during glycolysis process. NADH formed during glycolysis process is entered into mitochondria matrix for its oxidative phosphorylation. And the third assumption is that no other intermediate is consumed to get product. So no other intermediate is consumed in the whole process. So this is the third assumption. And the fourth assumption is that only glucose considered as substrate only glucose. So these are four assumptions for proper getting 38 molecules of ATP. So now let's read NCERT of this topic. So the respiratory balance sheet. It is possible to make calculation of the net gain of ATP for every glucose molecule which is oxidized. But in reality, this can remain only a theoretical exercise. These calculations can be made only on certain assumptions, and the assumptions are as follows. That first assumption is there is a sequential or orderly pathway with one substrate forming the next, and with glycolysis, tricarboxylic acid cycle or Krebs cycle, and electron transport system pathway. And the third assumption is that NADH synthesized in glycolysis process is transferred to mitochondria matrix for undergoes oxidative phosphorylation. None of the intermediate in the pathway are utilized to synthesize any other compound. only glucose is being respired, or only glucose is considered as substrate for this whole process. No other alternative substrate are entering in the pathway at any of the intermediary stage. So these are the assumptions for this theoretical exercise. But this kind of assumptions are not really valid. So these assumptions are for theoretical gaining ATP, but this kind of assumptions are not really valid in a living system. All pathway work simultaneously. All pathway like glycolysis, link reaction, Krebs cycle, and electron transport system, all pathway work simultaneously and do not take place one after another. Substrate enters the pathway and are withdrawn from it as. So substrate enter the pathway and are withdrawn from it as and when necessary. ATP is utilized as when needed, and utilized of ATP. Enzymatic rates are controlled by multiple enzymes, yet it is useful to do this exercise to appreciate the beauty and efficiency of the living system in extraction and storing energy. Hence, there can be net gain of 38 ATP molecules during aerobic respiration of 1 molecule of glucose. Now let us compare fermentation and aerobic respiration. Fermentation and aerobic respiration. So as we know, fermentation is an anaerobic process of extracting energy. So fermentation accounts only a partial breakdown of glucose, or partial breakdown of glucose by glycolysis process only. And after glycolysis, fermentation occurs, in which two types of fermentation: alcoholic fermentation or lactic acid fermentation. Whereas in aerobic respiration, it is completely degraded to CO2. So in fermentation, uh, there is partial oxidation of glucose takes place, but in case of oxidative or aerobic respiration, there is complete oxidation of glucose takes place, in which glucose is completely degraded to molecules of CO2 and H2O. In fermentation, there is net gain of only 2 molecules of ATP for each molecule of glucose degraded to pyruvic acid. So in fermentation, net gain of only 2 molecules of ATP by glycolysis, and for each molecule of glucose degraded to pyruvic acid, whereas many more molecules of ATP, like 38 molecules of ATP that we are extracting by the aerobic respiration generated under aerobic condition. NADH is oxidized to NAD plus rather slowly in fermentation, because there is no aerobic respiration, so there is no electron transport system or oxidative phosphorylation. So NADH is slowly oxidized to NAD plus in fermentation process. However, the reaction is very vigorous or fast. Now let's get started the amphibolic pathway. So respiration is an amphibolic pathway, means that in a respiration process, both process occurs, both process that is the catabolic one and the anabolic process. So basically in catabolic process, breakdown of molecule takes place, and in anabolic process, formation of molecules takes place. So in respiration, both type of process occurs, that is catabolic or anabolic. Like in respiration, glucose is breakdown to carbon dioxide molecules, completely break down to carbon dioxide molecule. This is a example of catabolic process. And in respiration, certain reactions occurs in which anabolic process shows. So that's why all the process of the respiration is amphibolic pathway, in which catabolic plus anabolic process shown. Glucose is the favored substrate for respiration. All carbohydrates are usually first converted into glucose before they are used for respiration. Other substrate can also be respired as has been mentioned earlier, but then they do not enter the respiratory pathway at the first step. So So it means that other than glucose, substrate are also being respired. That is fats, glycerol, proteins. These substrate are also respired for extracting energy, but they do not enter the respiratory pathway at first step of glycolysis. So let's see figure 12.6, that here the respiratory substrate is fat, carbohydrate, and proteins. So carbohydrate is simply break down to simple sugar like glucose, and glucose enter into glycolysis, converted to glucose 6 phosphate, and whole glycolysis ends to a molecule of pyruvic acid. Then pyruvic acid to acetyl coenzyme A by link reaction and then enters to Krebs cycle. But if we consider fat as substrate for being respired, so fat is two types, that is fatty acid and glycerol. So fatty acid and glycerol. So if fatty acid is considered as substrate, then it undergoes to direct acetyl coenzyme A and then Krebs cycle. If glycerol considered as substrate, then it changes to dihydroxyacetone phosphate and then glyceraldehyde and then enters into respiration. If proteins or we can say amino acid is considered as a respirate, so in case of protein, uh, according to its structure and its chemical nature, it enters into as pyruvic acid, acetyl coenzyme A, or directly into Krebs cycle. So entry of different substrate in the respiratory pathway. Fats would need to be broken down into glycerol and fatty acid first. If fatty acid were to be respired, they would first be degraded to acetyl coenzyme A and enter the pathway. If glycerol would enter the pathway after being converted to phosphoglyceraldehyde, the proteins would be degraded by proteases and the individual amino acid after deamination, depending on the structure, would enter the pathway at some stage within the Krebs cycle or even as pyruvate or acetyl coenzyme A. It basically depends on the structure of that amino acid. Since respiration involves breakdown of substrate, so respiration involves breakdown of substrate, the respiratory process has traditionally been considered as catabolic process. So due to this breakdown process, it is considered as catabolic process and the respiratory pathway as a catabolic pathway. But is this understanding correct? We have discussed above at which points in the respiratory pathway different substrate would enter if they were to be respired and used to derive energy. So different substrate is respired to derive energy. What is important to recognize is that it is these very compound that would be withdrawn from the respiratory pathway for the synthesis of said substrate. Hence, fatty acid would be broken down to acetyl coenzyme A before entering the respiratory pathway where it is used as substrate. But when the organism needs to synthesize fatty acid, so, but when organism needs to synthesize fatty acid, acetyl coenzyme A would be withdrawn from the respiratory pathway for it, meaning that fatty acid is converted to or break down to form acetyl coenzyme A. Acetyl coenzyme A. So this is a catabolic process. But if the body of the organism needs fatty acid, then acetyl coenzyme A, molecule of acetyl coenzyme A forms molecule of fatty acid, and this process is anabolic. So that's why we call respiration as amphibolic process. So there are many reactions. Hence, the respiratory pathway comes into the picture both during breakdown and synthesis of fatty acid. Similarly, during breakdown and synthesis of protein to uh, so similarly, in case of protein, respiratory intermediate from the link. Breaking down process within the living organism is catabolism and synthesis is anabolism. Because the respiratory pathway is involved in both anabolism and catabolism, it would be hence to better considered as respiratory pathway as an amphibolic pathway rather than as only catabolic one. Now, the last topic of this chapter is the respiratory quotient. So, respiratory quotient. So, respiratory quotient simply its formula is the volume of CO2 evolved to the volume of O2 consumed, or the respiratory wait a minute, let's look at this. So the respiratory quotient tells us what kind of fuel our body is burning. If it is close to 1, that volume of CO2 evolved upon volume of O2 consumed, both are equal, then we are mostly burning carbs. Then this respiratory quotient is for carbohydrate. If it is close to 0.7, means we are burning fats. Fats gives more energy than carbs when we burn them, but fats need more oxygen for its breakdown. So let us now look at another aspect of respiration. As you know, during aerobic respiration, oxygen is consumed and CO2 is released. The ratio of volume of CO2 evolved to the volume of O2 consumed in respiration is called the respiratory quotient or respiratory ratio. That is volume of CO2 evolved upon volume of O2 consumed. The respiratory quotient depends upon the type of respiratory substrate used during respiration. So it depends on type of respiratory substrate. When carbohydrates are used as substrate are completely oxidized, the respiratory quotient will be 1 because equal amount of CO2 evolved or equal amount of oxygen is consumed. As shown in equation that is given below here, that is glucose plus 6 molecule of oxygen is consumed and 6 molecule of carbon dioxide is evolved during this oxidation, plus 6H2O plus energy, that is respiratory quotient is 1. When fats are used in respiration, the respiratory quotient is less than 1. Calculation of fatty acid triple methane if used as substrate is shown. So triple methane is a fatty acid, uh, and it's here 145 molecule of oxygen is consumed to evolve 102 molecule of carbon dioxide. So its respiratory quotient is less than 1, that is 0.7. If when protein are respiratory substrate, the ratio would be about 0.9. So, what is important to recognize is that in living organism, respiratory substrates are often more than one. Pure proteins and it is important point that pure proteins or fats are never used as a respiratory substrate. So basically, Respiratory quotient of carbohydrate is 1, and for fatty acid like triple methane of fats it is 0.7, and for protein it is 0.9, and pure protein or fats are never used as respiratory substitute. So with this we ended this chapter. So thank you very much. If you feel any doubt related to this topic or this chapter, feel free to ask in the comment box. And if you like this video, don't forget to hit that like button. So thank you very much. Bye bye.